Hello everyone, I'm Soumya, working as an assistant professor, Department of Zoology, Government First Grade College, Vijayanagar, Bangalore. In this video, I'm going to explain locomotions in case of protozoans. In case of protozoa, we can find different varieties of locomotions, that is the pseudopodia, flagella, and the ciliary modes of locomotion will be seen in case of protozoans. So protozoans, as we all know that they are the microscopic unicellular organisms, exhibit different modes of locomotions. In case of the pseudopodia, pseudopodia are nothing but the temporary outgrowths of a leg-like structure. Pseudo means the fall, podia means the leg. So the pseudopodias are temporary outgrowths of the protoplasm from any part of the body. Here in these individuals, the body of these individuals is usually not covered by any hard exoskeleton. Uh, or else it will be supported by means of a pellicle sometimes. Usually the pseudopodians will be formed out of the movement of the ectoplasm and the endoplasm uh, towards the formation of the pseudopodia. So based on the shape of the pseudopodias will be differentiated into four different types, that is lobopodia, philopodia, rhizopodia, and axopodia. Well, yeah, these are the four types of pseudopodias that will be seen in case of the protozoans. So the first one is the lobopodia. Lobopodia is nothing but it's a blunt, short, finger-like projection that will be produced by the protozoan towards the direction of motion. So whenever they want to move, it produces a blunt finger-like projection. That is what we call the lobopodia. The best example for these are the amoebas. And whatever the finger-like lobopodia that produces in these individuals, it produces based on the movement of ectoplasm and endoplasm. Yeah, that is the lobopodia. Next comes the philopodia. Phylopodians are the one, these are fine thread-like structures. Usually will be having the rounded ends at the tip. So these are sometimes branched along with having a finger-like fine structures. They'll be branched somewhere sometimes. See, this is how they'll be branched. And sometimes these are interconnected. Here, these are produced or these are made up of hyaline ectoplasm. Only the hyaline ectoplasm is involved in making the phylopodias. So example are the radiolarians. Usually they'll produce these kind of pseudopodias, that is the phylopodias. Next is the rhizopodia. Yeah. Here it is the rhizopodia. Rhizopodias are, or it is also called reticulopodia. So the reticulopodias are also thin, long, branching structures. Usually they'll be present adjacent to each other. So these reticulopodias usually they'll not form because of locomotion. Usually these reticulopodias will be formed in order to capture the food. So those are the reticulopodias. Next is the axopodia. So the axopodians are these are semi-permanent. So as I was explaining the pseudopodia, I said it is a temporary structure. But in case of the axopodia, unlike the other pseudopodial structures, these axopodias are semi-permanent structures. So the axopodias, these are quite long, stiff, thread-like structures, which is formed out of the ectoplasm with a hard central axis. See, we can see, well, where's the mouse pointer? Yeah, these are the axopodial extensions. The axopodial extensions are supported by means of a long central, stiff central axis. So that's why these are semi-permanent. So the axopodial here also, the function of these axopodials majorly not for the locomotion, but in order to capture the food. So this will be seen in actinophytes. Yeah, this is all about the pseudopodias of protozoans. So next we'll move on to the flagellary motion. So the animals which possess flagella are flagellar individuals. Flagella is nothing but vipe-like structures, here thread-like structures. See, flagellas, when you look into the fine structure of flagella, so the flagellas will be having the central microtubules. A pair of microtubules will be there, and that will be arranged in the, see, here in this, we can see the inner microtubules. So this is how the cycle wheel and the spokes like arrangement will be there for the microtubules and the flagella. So inner microtubules are there and outer doublet microtubules are there. Usually these are 90 numbers. Even this kind of structure will be seen in case of the microtubules, in case of the higher vertebrates. So here there will be continuous arrangement of these microtubules, central and the these outer microtubules. So they'll form, so this is how the polymerization or the continuous arrangement of these microtubules forms a thread-like structure. So this is how ultimately the flagella happens because of the continuous arrangement of these structures. So ultimately there will be 20 fibrils are there and 20 fibrils arranged in a manner and they'll be compressed in a structure in the cytoplasm. See, here it is the plasma membrane which covers the cytoplasm, and within the cytoplasm structure we can see the presence of the microtubules. So the 20 microtubule fibrils have been arranged in a structure and forms a thread-like structure, that is what we call the flagella. So the flagellas are usually thread-like structures. So flagellas, they'll be provided with the mastigonemes. So these hairy-like structures are mastigonemes. So along with that, they are provided the filament. So the based on the presence of the mastigonemes, we can categorize the flagellas into various types. Take for example, the first one is the uh, stichonematic. So in case of the stichonematic, the arrangement of the mastigonemes will be on one side. See, you can see the mastigonemes have been arranged in one side of the flagellum. So then it is called a stichonematic. Stichonematic is usually seen in case of the euglenias. So next is the pentanematic. In case of the pentanematic, here also the flagellas provide with the mastigonemes, but the mastigonemes are arranged in both the sides. So in the flagella, if both the sides mastigonemes are present, then it is a pentanematic. The next one is the acronematic. In case of the acronematic, we don't find any mastigonemes. Instead of mastigonemes, the terminal end of these flagellates will be having the terminal filaments. So that is the acronematic. Next is the pentacronematic. In case of the pentacronematic, along with the terminal filament, they'll be having the mastigonemes, but that mastigonemes have arranged in a pattern that they have been arranged in sideways. That is pentacronematic. And the last one is enematic, or otherwise it is called a simple flagella, where these individuals, these flagellas, they'll be not having any mastigonemes or any terminal filaments. So that is enematic or simple flagellas. So based on the presence or absence and on the arrangement of the mastigonemes, will classifies the flagella into stichonematic, pentanematic, acronematic, pentacronematic, and the enematic or simple nematic. So those are the flagellary structures. Next, we'll move on to the cilia. So the best example for ciliary modes of locomotion is the paramecium. Here we can see the paramecium with the cilia, presence of cilia. When we look into the inner structure of the cilium, same as the flagella, cilia are also possess the central microtubules and the outer microtubules in the same pattern. But the thing is, cilia are small hairy-like structures. Sometimes they'll be not connected with the nucleus. So usually in case of the primitive organisms, the cilia will be covered entirely to the body. Whereas in case of the higher individual, ciliary structures will be restricted to certain sites in forms of bands. In case of the verticella and all, the ciliary bands are there. So that will be seen in case of the higher protozoans, where the the cilia will be restricted to certain band or filament-like structures. So this is all about the locomotion in protozoa. So to summarize the whole thing, in this video, we discussed the locomotion, various modes of locomotion in protozoa, that is the pseudopodia, flagellary and ciliary locomotion. And one more thing, the myonemes. Myonym locomotion will be seen in certain protozoans like the entomibas, even in the euglenias. We'll see the myonym locomotion along with the flagellary locomotion. So in case of the myonym locomotion, there will be a continuous movement of the cellular materials within. It is like a peristaltic movement of the intestine. So with that movement, the continuous movement of the components within the cell leads to the movement of the protozoan. So the pseudopodial structures are the temporary finger-like projection. Based on the structure of pseudopodia, we'll classify it into lobopodia, axopodia, reticulopodia, and gyropodias. Then the flagellary structures are there. Flagellas are white-like structure. Based on the presence or absence of the mastigonemes, we can classify the flagellas into stichonemata, pentanemata, acronemata, and pentacronemata, and the simple flagellas, and the ciliary motion and the myonemes. So these are the various protozoan motions that we discussed in this video. And these are the references I referred for the particular content. Thank you.